Yeah, okay. Yeah, it's fine. It's fine. So they are related, but it's not. So the one on the left is related to the microfilm. The microfilm has a bit still different morphology. They project the jaw, and they. Yeah.
Oh, the one
No, so that's a cichlid fish, which is actually from deep Prague, but not from the beach. So I am like comparing it with my garden state. And it's
no, no, 1 hour in total.
yeah, no, done. So should I try to be really done with it?
Yeah, I will consider that we start later. So yeah.
Right. I have several.
No, perché questo qui è un altro.
Ma non sappiamo cosa esiste perché già una volta uno per 10 studenti, interessante. Non lo so vero se bravo, hai fatto bene, sì, dato okay. Okay, okay, so
Not sharing, but I don't know, even recording.
okay. Good morning to everybody. So it's a pleasure for me to have here Professor Zuzana Musilova from the University of Prague. She will tell us about her research and something that we are recently interested in as well, so the biology of organisms in the deep sea. So just a brief introduction, she did her PhD at the University of Prague on fish biology. Then she moved to the University of Basel for her postdoc in the lab of Professor Walter Salzburger, that was a guest of Stazione Zoologica a few years ago as well. Well, very briefly, she got the L'Oreal Prize for Women in Science in 2020, the Neuron Prize for Young Scientists in Biology in 2021, and very recently and importantly, the ERC Consolidator Grant. So welcome and please.
Okay, thank you for the introduction and thank you for the invitation. So I'm happy to be here. Yeah, okay. Right. So I'll try to speak close to the microphone. Yeah, so I'm happy to be here to tell you about our research on deep sea fish and also other a bit of other deep water fish, but mainly it will be deep sea fish. And yeah, how exciting they can be. So I'll be talking mainly about the deep sea fishes, which you can see they look, sorry, I'll try to get rid of this thing. I'm trying, no, right.
Okay, I will.
Yeah, okay. Yeah, because all the titles would be covered otherwise. Thank you. No, so I'll be talking about deep sea fish. I will also have a couple of slides on the deep water cichlid fish from the crater lakes in Africa. So they're freshwater fish which live in the volcanic lakes, and they dive in the deep, and they live there. So it's like a parallel to the adaptation to life in the deep water. And before I start telling you about the research itself, I would like to say that all of it was possible thanks to the collaboration with many research teams all around the world. Yeah, so the point is that it's not here. Yeah.
Devi uscire dalla presentazione.
Right. Okay, now I see. Yeah. Seems to work fine. Okay, thanks. Anyway, so yeah, just to conclude that, so it's been possible thanks to a collaboration with different research teams from all around the world, because what you will see, it's not in the capacity of one research team to do. There are very different things. And once you have your own hypothesis, you want to get it tested with the better or the best possible way you can imagine, and usually you need to get in touch with specialists. So big thanks or acknowledgement to these people. I'll still have the final acknowledgement, but this is to open my talk with. So if you dive in the deep sea, it itself is an environment with extreme conditions. So of course you have the enormous pressure there. Sorry, you also have low temperatures there, even in the tropical regions. And there is like low or no light. This video I'm showing here just to introduce you the environment of the benthic deep sea. And those are the deepest filmed fish ever, which are from 8.3 kilometers from last year, April. So this is like what you can see at the bottom of the sea in the very deep. If you are in open water, in the not so deep, but still a deep sea, you can see a bit more bizarre creatures. So if you look at the deep sea fish, the mesopelagics, or those which are swimming in open water, they look really, some of them very bizarre, and they have like the extreme adaptations, like huge eyes, or you know the big teeth sometimes. And all of it is to cope with the difficult or harsh conditions down there. And we will be talking about vision here today. So the extreme visual adaptations we find in the deep sea fish are basically to cope with the almost dark conditions. Some of the species look very kind of noble, or you would call them like majestic, but also it depends on the angle of the way you look at them. So the same species also may look like this. So this is just a different specimen of the same species. So yeah. If you are a deep sea fish, you kind of have the same to-do list as all other organisms on Earth, which is basically to find a prey, to feed, to find a mate, to reproduce, and to avoid predators at least for some time, so that the life can go on. And all these tasks are, of course, dependent on the sensory systems. And when you're in the deep sea or in any other extreme conditions, if your sensory systems or any of your sensory system is enhanced a bit, so it's better, you have a huge advantage over the others. So that's what drives actually the evolution of the extreme adaptation of sensory systems. I will be talking about the visual system, so vision, the sense of sight. And the adaptations can be found at different levels. You can see it already from the far that there is adaptation on the eye size or eye morphology. So if you look at some of the deep sea fish, their eyes look big. If you would zoom in a bit inside the eye, you also see adaptations in the retina anatomy. So you would see multiple layers, or like super long rod cells and so on. Or if you zoom even more inside the photoreceptor cells, you find the adaptations on the molecular level, like in the photoreceptor cells themselves. And this is what I will be talking most. But let's have a quick introduction, a quick start with the simplest adaptation to low light condition, which is in the eye size and morphology. I already mentioned that the deep sea fish live at different depths, and you indeed have different zones in the deep sea. And there is a zone called mesopelagic, which is between 200 and 1,000 meters of depth. And this is a zone where you still get some remaining of the sunlight coming from the surface. So there is still light from above. And below 1,000 meters, like all these deeper zones, there is no sunlight coming from the surface whatsoever. So basically no photons which would be detectable by any biological system. So we also make sense that most of the fish living here in this mesopelagic zone are those which have the extreme adaptations. If you live down here, perhaps it doesn't make too much sense to enhance your vision because you don't use it that much anymore. And also, if you look at the deep sea, this is the zone where you have the highest abundance and highest species diversity of all deep sea fishes. So if you simply compare the relative eye size, so basically the eye to the head size, and you plot it against the depth. So here is a depth, here is the eye size. You really see that what happens mostly is between 500 and 1,000 meters, and then it really drops down. So fish living in the 3,000 or 4,000 meters depth, they have relatively small eye size. So they still have sight, they still have vision, but it doesn't make sense to invest much in the huge eye. It's just fine to have some eye, because when you are below 1,000 meters, you can see some bioluminescence, but not environmental light. And now let's talk about some morphological adaptation, which is quite extreme. So the first and easiest thing what you can do if you want to adapt is to simply increase your eye size. So that happens to many deep sea fish. So they have simply bigger eyes. But at some point there is a limit which you cannot go over, and that's the size of your head. So you can't increase the eyes infinitely. So if they don't fit in your head, there is still a little trick of what you can do. And this is to omit the side parts of the eyeball. So you can increase your eyeball even more, but you get rid of these side parts and you just keep the central cylinder, which is the most functional part. Then you can still increase it a bit. And this is how the so-called tubular or barrel eyes have evolved. And you may then look at them here. So you see that it's a huge lens, and here is the cylinder. So if the eye was complete, the eyeball would be this. But it's not, so it's only the central cylinder, and you see how it still fit in the maximum size of the fish head. So this is how it may look from when you look at the fish from the front, and these are the tubular eyes. And by default, the tubular eyes are actually oriented upwards, because many deep sea fish they are predators, and they work the way that they look upwards and they screen the remaining light for the silhouettes of the prey. So getting the silhouette of something above you, that's the most common way how you as a predator capture your prey. So most of the fish in the deep sea, they look upwards and they screen for these silhouettes. If you look at this fish, here the green balls are the lenses. Here is the barrel eye looking upwards. These things here, these are nostrils. So this is a nose, not eyes. Don't get confused. And a little mouth. Here is a drawing how it looks. And this fish has been filmed recently by the Monterey Bay Aquarium Research Institute. And it was quite surprising, because it was for the first time where they could see that there is some transparent layer covering the eyes. So you can find these videos on YouTube. And if you go to the MBARI, or Monterey Bay Aquarium Research Institute, then you will find many other deep sea fish. They have really cool collection. And look at this fish. So the fish is looking upwards. Here you see the huge lenses, and the fish basically screens what's above. And you will see they will also try to capture it towards the end of the video, because they specialize on capturing deep sea fish and bringing them alive on board. So they have a special device which you will see, which can capture the fish and close it and then depressurize it gradually. But the tricky part is to get the fish in, obviously. So this is what they try. There is one, the barrel eye fish. Here is the device. They try to slowly get under it and kind of scoop it. And remember that the fish is looking upwards. So it doesn't see what's happening below it. So now they test the lid and now they go slowly up to get the fish in. So the fish still doesn't know something is going on, still doesn't know, and now it realized. So it escaped in the very, very last moment, which is a bit sad ending of this video. But yeah, still they keep doing this. So sooner or later they will get one up alive. And what I showed you is also illustrates the side effect of getting improved vision. So these fish, they look upwards and they have really perfect binocular space allowing or distance allowing vision when they look upwards. So this is what they have. Binocular vision angle is here. The problem is that they don't see anything below them. There are some morphological adaptations how they try to improve their angle. So you may get some reflective tissues still in the retina, in the eye to widen the angle a bit, which happens. So the angle is widened, but still it does not solve the situation that there is like a dead angle under the fish. There are species related to this barrel eye fish that have overcome this issue. And it's perhaps the most extreme morphological adaptation that can be found in the deep sea fish. And it's called mirror eyes. And it's a type of eye where the big tubular eye is still oriented upwards, looking the same way as we showed. But there is a little tiny eye on the side, which is open in the direction to the downwards. This eye does not have lens, but it has a little mirror from the guanine crystals. And the flashes of bioluminescence coming from below, like imagine from here, they just reflect here and go on the accessory retina here. So the fish can detect also some flashes from what of something what's happening under it. So these this is like the most crazy or the most extreme morphological adaptation of the eyes among vertebrates. Here you can see it how it really looks. So if you look to the fish from above, you see the two lenses and like two side eyes. If you look inside the side eyes from the bottom, so from the ventral view, you see the opening here. The white tissue is actually the accessory retina. So yeah, so this is like the evolution of the mirror eye in this group it's like the most advanced, and this is one of the extreme adaptations you can find in the deep sea. I'll now jump or zoom in the photoreceptor adaptations, which I'll be talking from now on. And before doing that, I'll just take you like to the zoom through the eye. So this is a camera type eye where you have the photosensitive tissue called retina. This is the same for every vertebrate basically, so including us, we have the same. So the camera type eye is the conserved structure. There is a retina, and in the retina there are photoreceptor cells. There are cones and rod cells, and these are actually responsible for color vision or for vision. So the cone cells are usually responsible for color vision. In human, it's the red, green, and blue photoreceptor, and the rods are responsible for low intensity vision, so in our case for night vision. And what makes the cells sensitive to the light is the photopigment, which is composed of the opsin protein and retinol. So, okay, yeah. the opsin protein and the opsin protein can differ in some amino acid sites, which then can change a bit the color sensitivity of the opsin or of the photopigment. So this is the protein, like the visualization, like a schematic. If you then want to zoom a bit in the rhodopsin protein, you can see here the drawing where you have one amino acid next to each other. So the whole protein is composed of 300, almost 50 amino acids. And there are some amino acids which are essential and which are known that if you change them, they change the color sensitivity. this is what also stands behind the evolution of human color vision. So in our blue, rod, and green cells or cone cells, we have three different opsin types, and they are actually different in these key sites, and that caused the shift of the sensitivity. So just by the DNA mutation, the protein can differ afterwards in the function, which in terms of in case of opsins means different color sensitivity. Now I'll briefly show a slide which I introduced to the students already in their presentation, but that illustrates basically that the number of the opsin genes and the number of the different cone cell types actually determines the vision. So we humans are mostly trichromatic, having the three channels, the blue, green, and red. And there is a photo of frog, which is red, which is green and has red eyes. And this is how trichromatic people see the frog. Some people lost one of the channels, the red channel. So some people have the blue and green channel only. We call them colorblind. They are not colorblind entirely. They still see colors. They're not able to distinguish the red and green. So if you look at the frog here, this is how the dichromatic people see the same picture as the trichromatic here. So if you see the two frogs the same way, it means that you are actually dichromat. And so this was to illustrate that, like, adding one more color channel by adding one more opsin gene type can actually mean a huge advantage for the holder or for the organism who has that. And this stands behind the human color vision. And most of the mammals, they do not have the three channels. They have only two. So most of the mammals are actually dichromatic. So your cat or your dog, they both cannot distinguish the red from the green. And this is how they see basically your world. So like, we humans are actually exception together with some other primates to see in the three color channels. So here is just a overview how it can look. So the trichromatic is our common day vision, where you distinguish the red from the green. The dichromatic vision means the lack of being able distinguishing red from the green. And the monochromatic is actually the black and white vision, which we have at night, because we have only one rhodopsin channel or one rod type at night. So at night, we all see actually only black and white. So that's the monochromatic vision. And yeah, I told you that humans have three cone gene types, three cone types, and then three color channels. This is easy. In fish, if you look at fish genome, you usually find more opsin genes there. So the ability of producing more different color channels. But it does not mean that you would have 6 or 7 color channels there at the same time. That would not be possible for the brain to process that. But the material in the genome is rich. So in some cases, the only subset of the genes can be used. But anyway, so fish in general have more opsin gene types than humans do. And also, if you look at throughout the diversity of fish, you'll find very different visual systems. So, but how is it in the deep sea fish? Because they are kind of, that's a weird environment where there are certain limitations. First, I'll tell you some kind of introduction to the genes, or just the introductory slides to the genes behind the vision. There are 5 different families of opsin, visual opsin genes in vertebrates. 4 of them are responsible for the cones or for the color vision during the day. And there is only one type of the rhodopsin, which is in the rod cells at night. This is shared across all vertebrates, and all the visual opsins are belonging to some of these families. Yeah, and as I said, the cone opsins are for the color vision during enough light. The rhodopsins are for the dim light vision. What happens with the light in the deep sea is that not only the intensity of the light is low, but also the spectrum is narrower. So both edges of the spectrum are not present in the deeper zones. And if you go deep, then you basically find blue and green lights only. So if some of you is scuba diving, you perhaps, or you can actually notice that already on your own, because already in 10 meters, you can see really lack of the red light. So if you look at something red in the deep, it won't look red at all anymore. So this is already the gradual lose of the red color in the depth. So it's a property of water filtering out these edges of the light spectrum. So we were interested in the opsin genes in the genomes of the deep sea fish. That's some time ago, and with my colleagues we screened the genomes of 100 fish species back then. And here you can see the phylogenetic tree of the fish species, with the dark branches being like more like the deep sea, while the gray means shallow water. So we were interested to see how many opsin genes are there. And first we looked at the cone opsin genes, and we were asking, like, do deep sea fish from different evolutionary lineages have some kind of similar adaptation or similar setup of the opsin genes? And yeah, so they are like at different places of the fish evolutionary tree, and we find some shared similarities. And this is basically the lack of the UV sensitive and the lack of the red sensitive opsins, which really makes sense given that there is no red or UV light in the deep almost. So this is something which still makes sense. It's not super surprising, but it's very shared, like very common in different lineages. I'll now make a short detour to another system, then we come back to the deep sea fish. Because something similar happens also in different fish adapted to life in the deep. And we also study the crater lakes in Cameroon, which are like deep lakes, small but deep, the volcanic lake. So this one particular lake has over 100 meters of depth. And there is a group of fish which has evolved there. Is a case of like small adaptive radiation, so something like similar to what you know from Malawi or Tanganyika or Victoria lakes, but in much smaller case. It's just one small lake, and there are only 11 species endemic. It's still a lot if you are there at the lake, which you can swim over, and there are 11 endemic species which are nowhere else and which have evolved there. But it's small, and like small parallel to the Malawi system. And the story is kind of similar. So they have evolved from one ancestor who colonized the lake, and you can see the same as what you see in the other adaptive radiation. So the shape of the mouth and the specialization for different food types, and also to specialization to different habitats. And two of the species have actually colonized the deep zone of the lake. All of the others are found together in the shallow water, but two of them are found in the deep, either only in the deep or seasonally in the deep, but both of them are from the deep lakes. They are also adapted in different other physiological features. So those living in the deeper water, there is like a very low content of oxygen, so they have adapted also in their hemoglobin genes, which has several mutations to cope with the hypoxic conditions, but we were interested in the vision. So we looked at the visual system of these cichlids, and in the genome they all have 7 cone opsins, so belonging to these 4 families. So that stays the same. It's just some of the genes are duplicated within the categories. And when you look at the genome or sequence level, is basically almost identical. So there is no difference between the 11 species and the genome or gene sequences. But if you look at the expression profile, this is what differs a lot. So here are the expression profiles, which are split in the single and double cones. That doesn't matter. So that's like a types of cones in the retina, but the proportion of color basically means which color channel dominates. So you can see that the shallow water, so the 9 shallow water species have kind of similar expression profiles, and they are at least trichromatic. Some are perhaps tetrachromatic. They are able to see in colors, having definitely a red and a green and a blue channels. and with the vision perhaps similar to ours in terms of color distinguishing. So that would be like the expression profile if I simplify it on this plot. But then just to remind you, the conditions of the light in the deep sea and in the clear oligotrophic lakes is similar. So also here, the attenuation of the red and UV light is present. And if you look at the expression profiles of these two deep water species, you can really see the lack of the red vision. So there's no red vision, and there's also no like the violet or blue violet vision. So only one of the central blue opsin is there. So they really lost the expression of these genes because they don't need them. So kind of, if you draw this, this is how the deep water species profile looks like. And then if you compare these two, you can see that functionally it's very similar. So they lost the vision in the UV and the red part of the spectrum. In this case, by losing the genes, because it's longer evolutionary time, so there was time to lose the genes. While here it's very short evolutionary time. It's like perhaps 100,000 years, no more. So the genome is still the same, but they lost the expression of the genes. So the loss of the expression might be way faster than waiting for mutations in the genes. So in this case, two different systems lead into the same functional outcome, just by very different way. So that was a detour. Let's go back now to the deep sea fish again. And yeah, here again, there are some photos. And so far, I've been talking about the cone opsins, those known like for color vision. But we are also checking and interested in the rhodopsin genes, even though there's only one family or one type of the rhodopsin in all vertebrates. But, and that's also the case. So most of the vertebrate species, including us, we have only one copy of the rhodopsin gene in the genome. And you can't do much. If you have one copy, you have one rod type, there is one color channel, no more. So that's a black and white vision. In some fish, there are actually two copies of the rhodopsin gene, and one of them is eel. The eel actually changes the expression of the one and the other copy to better see during the migration. One copy is for the freshwater phase, one is for the deep sea phase. So there are some advantages of having two copies. But how is it with the deep sea fish? So we looked at the genomes and we screened the genomes, and in most of the cases you still get only one copy of the rhodopsin, but there are 3 particular cases where the rhodopsin gene has been duplicated. And the extreme is this species, or actually 2 species, which have up to 38 rhodopsin genes. So this is something very unusual, and it was something like, wow, so what's going on? So having 38 rhodopsins was back then when we found it, like most of all animals. Later, some shrimp was found with 40. So like it's not the most, like the highest number among animals, but it's still the highest number among vertebrates. But the number itself doesn't, like, it's not that interesting, because you can imagine that you may have many genes, but if they are the same, they just produce more product, but the product would be the same. But in this case, it's not the case. So the rhodopsins of these genes, they differ a lot in the function, so that they are actually covering the entire light spectrum in the deep, so from the blue to the green, and you can see how like tightly it's like one next to each other. And we don't know the function of this system yet. So, but it's really something unusual and like unseen elsewhere. Just to give you an idea, I'll project it now over our own visual system. So this is a human visual system, which you have seen already, the 3 cone cell types. And if I project the spiny fin, so the fish with the highest number of rhodopsins on top of it, you see that all of the sensitivities are actually between our blue and green. I mean, would be a nice complement to our vision. Anyway, so this is something like also quite hard for us to imagine how exactly they can see. And it's true that we don't know. And if you look at the spectra of the residual daylight, so what's coming from the surface, or the bioluminescence. Both of them are lying actually in this sensitivity of this fish. So both of these are definitely being seen by this fish. And the question now is why and how. So how does it work? Like, we don't know, but we try to find out. One of the options is that they could perhaps have some kind of color vision that would be useful maybe to discriminate the bioluminescent prey of different lights. Maybe that could be, if they are specialists, which we don't know yet, then seeing like your prey would make sense. If that was a case, then that would be the first case when the color vision would be mediated by rods, which is also something the vertebrate eye was not thought it would be possible. And to answer that, we would need to go to see the expression in the eye. And we already know the total RNA expression. So there are like over 14 or at least 14 different rhodopsins. I'm showing here just 3 of them to simplify the figure, because I'll continue with this little pie chart. But imagine there will be 14 different categories. So for at least 14 of the 38 rhodopsins are used in the adult fish. So they are using it. It's in the retina and it's there to see. But yeah, how exactly it works, we don't know. Yeah, sorry, I have the old conference name there. Anyway. So what we want to do and what we are now about to do is to look at the single cell level. I mean, that gives you an answer. How is it at the single photoreceptors? So the two options are that you either get everything expressed at every cell, so all the cells would be the same, but they would be super sensitive because they would be able to see every color. Or you would get some specialized cells, which would some would be in more green, some in the more blue shade of the light spectrum. And this is basically the thing you can answer with the single cell approach. So are there like some super powerful rods with some kind of wide spectrum, or are there like the different color channels kind of, which could then enable the color vision? We don't know. And we will, so that's why we will do the single cell. And yeah, this is also what my my ERC grant is for. So from now on, we have 5 years to really work on these deep sea fish. And I hope we will get the answer, because it's not that easy to work with the deep sea fish. I can tell you afterwards if you want. Anyway, I'm going back to the rhodopsin spectrum, because I mentioned in the beginning that in the rhodopsin there are certain amino acid sites, here highlighted with the yellow, or by the yellow color, which are important, or which are known that if there is a mutation, they will change the color sensitivity of the rhodopsin. And the first thing we did with the 38 rhodopsins was to check these sites. So are they different? Before you do any functional test, you actually can, or you want to know if there are differences in these sites, because that's a good estimate that there will be functional difference. The sites are listed here by the number. The number doesn't matter. It's just like corresponding to the reference of the cow rhodopsin. And we checked which of them are actually variable in this 38 spinefin rhodopsins. And turns out that it's actually most of them. So if you like look at this, it's just like, wow, so there will be sort of like definitely functional differences. And then afterwards, when we did the combination of approaches, we ended up with this plot, what I've already showed you, so that it's really, really functionally differentiated. So we still don't know what is it good for. And I mentioned that bioluminescence around them might be one of the things. And if you look at the bioluminescent spectra in the deep sea, which is this light blue color, you see that this is all organisms there, not only vertebrates, so invertebrates, vertebrates. How many species do have like the bioluminescence in this wavelength of color, so like the bluish type? And it's really dominated by the wavelength like between 460 and 480 nanometers. And if I project the sensitivity of the spinefin over this, you can see that it's also in the middle of their spectral sensitivity. So it may be serving for that. But yeah, as I said, we actually do not know very well. So I said something about this species, which has 38 rhodopsins. This is the extreme, but have in mind that the same thing has evolved also twice at two other lineages in the evolution. So they are completely unrelated to each other. And here the same story happened. It's just with the 6 rhodopsins. Here the same story happened with the 5 rhodopsins. And the thing is that even if we wouldn't find this one, which was found just by coincidence, because the selection of species was random in the beginning of the genome scans, even this would make the story. So even here, the situation is kind of similar. These 6 rhodopsins are different and are covering the spectrum. It's just here is way more exaggerated to the extreme. So that's why he took all the fame, while these 2 are almost mentioned. But just have in mind that the same thing has evolved 3 times independently in parallel, because the deep sea conditions are really favoring, obviously, this adaptation. But why? So the question from the other side might be: so why do we see this and not like every deep sea fish having the same thing? That's also a good question, because we don't know the answer to that. But the gene duplication is obviously not that common. So in many lineages, it just hasn't happened. And there are even some cases of deep sea fish which would really benefit of having multiple rhodopsins, and which have been speculated that they must have it because of some functional peculiarities, but it's just not the case. So these guys, they do have it. They are somewhat lucky that in evolution it has happened. So yeah, so my point at this part of the talk is that we don't know how they perceive their world. Maybe they see really full colors, like their brain makes the full colors out of what is down there. We don't know. And now I'll have a still like small final or closing little story about the deep sea fish larvae. So far I've been talking about the adults, and I've told you something about the deep sea fish life, like how they see, how they catch fish, but not about the larvae. And it's actually a funny thing, or like not so funny, but like the many deep sea fish, the mesopelagic, they are, even if they are deep sea, their larvae start in the shallow. So they spawn in the deep, and their eggs, they go up, and the larvae are in the shallow water. So you can go and capture them in the shallow water. And they are there living for some time. And after that, they dive down to the deep again. And then some of the species stay down there forever. They never come up back. Some species do migrate on a daily basis, like at night up, but that's already a species specific thing. But in the case of spinifin, it's not a migratory species, so it does not migrate to the shallow, but the larvae can be found in the shallow water. So obviously, there is a time window in the deep sea fish when when there is enough light. So maybe the cone cells could be used even if they don't have them as the adults. Who knows? So we are also interested in how it looks in the larval vision. And I had a student who did his PhD on different deep sea species, and he compared the larvae and the adult individuals and the expression of the eye. Yeah, here Nick, and he found out that in different fish lineages, here again, those are unrelated deep sea fish lineages, the larva always has a cone and the adults have the rods. So in many species, you find only rods or rhodopsin in the adults, but the larvae always have some conopsin. And in the case of most of the deep sea fish, it's the green sensitive conopsin. Why green? It doesn't matter, but it's the green which then is sensitive to the green and blue. So it corresponds to the kind of to the middle part of the spectrum, but it differs drastically. So you can see that like you may have even complete replacement of the opsin type. And well, if you look at the fish here, so this is the larva of the fish, which is here as an adult. So you can see that I think it just died. No, it's there. So they have eyes on the stalks. So something very, very unusual. So another a crazy adaptation. So the larvae of one of the species of the deep sea fish has eyes on the stalks, while the adults look very different with the large teeth and the bioluminescent barbel. So there is a huge like morphological change or metamorphosis from the larva to the adult at any like many other aspects. And it also includes the composition of retina. And yeah, he also did a bit more detailed developmental series with the fang tooth. That's like one of these iconic deep sea species, because we had the chance, we were lucky to get the samples. So so he could do it, and you can actually see the complete the developmental switch from the green, which is cones, to the rods, which is shown by gray here. So it's really a gradual change. And our question was, so how is it with the spinifin? So the one which has now the 38 rhodopsin gene. So what do we see there? And this is the expression of adults first. So let's just go, because we're going to compare it. So each of the letters on the x-axis is the type of the gene, and they are ordered from the most blue sensitive to the right to the most green sensitive. And always there are 3 columns, is the 3 individuals we tested back then. So the expression is kind of across the entire light spectrum there in the adults. If you look at the larvae, it's very different. So you have one of the shortest and one of the longest sensitive ones. And note also that it's the bars are even cut, so they would be much higher. So it's really like an extreme difference. So the one of the shortest and one of the longest sensitive rhodopsins are there. But there is nothing in the middle. And it's because it is complemented also by the cone opsin here. So there are still some cone cells in the larval retina, obviously. And so it's combination. So even as a larva, this species like already takes advantage of having some differentially sensitive rhodopsins. So it combines like the cone cells, which are normal larval genes, with the short and long sensitive rhodopsin. So here again, the vision is obviously enhanced. We don't know how, but it's like taking advantage of having multiple rhodopsins. And now I will basically close my talk by saying, or by summarizing what I talked about, but also like saying the thing that we know a lot about this species. So this species is actually is actually one of the species with the largest eye diameter. So if you remember the plot with the eye diameter, he was one of the top. It also has the very thick retina. In fact, like one of the thickest retinas known. So you can stick the needle and it stands. So it's really thick. It also has super long rods, which contributes to the thickness of the retina. So that's together. It has the highest number of rhodopsin or of opsin genes. So 38 rhodopsins plus 2 cone opsins. So it has 40 visual opsins. We also know that the opsins functionally, or the rhodopsins functionally cover like the ambient light and also the bioluminescent spectrum, and that the larvae are combining the expression of the longest and shortest sensitive with the cone opsin. So these are quite some things we know about a species, but all of these like deep sea samples we get, they come from the deep when you are on board like a vessel and you deploy the net and then you get the samples. So no one has ever seen this fish alive. So there's no footage, no video recording. We don't know how it behaves. We don't know almost anything. All we know is from the samples we get on board of the dead fish. Well, it's got even its own beer brand, turns out, but still, no one has seen it alive. So this is the reality of working with the deep sea samples. And A, like no one has seen it alive. B, is super difficult to get samples. So all of what we did so far and what I told you was based on one individual sequence for the genome and 3 individual sequence for the transcriptome. But we were not lucky to get fresh samples after we found out. And since then, like this species is on a wish list of several deep sea cruises. I'm still waiting to get more. So 2 of my PhD students, they went on a cruise last year and they were lucky to get 1 individual. So we were super happy. So now we can get the high quality genome and we fixed the eyes for the single cell approach. But of course, because you have 1 individual, only 2 eyes, you wanna be sure that your method is working. And that's the reality. So you cannot get 10 replicates, which would be great, but we just don't have it. So yeah, so with that, I would like to end my seminar. This is my busy acknowledgement slides mentioning the members and the funding and the collaborators. And yeah, I'm happy to get being contacted by anyone in case you have further question or some looking for positions. And yeah, I thank you for your attention.
Thank you very much, Zuzana. Really inspiring. And yeah, there is time for few questions.
Hi, thank you very much. Very fascinating. I was wondering if you have, as you said, it's very difficult to have samples, so probably it's a naive question, but if you have an idea of the composition of the percentage of rods and cones, or if you want to look for that, because of course, as we saw in vertebrates, there is different percentage patterns that also are relevant for the visual.
Yeah. So this thing is quite easy to be checked even on the dead individual.
Okay.
So you can open the eye, dissect the retina, and then check the percentage. And it's been even done in the past. So we know the composition in this particular fish is mostly rods. There is a little patch also of cones, but that might be like the helping to see a bit better in case there is like some light coming from the surface, but most of the retina is composed of the rod cells, and they're actually different. I might even show you. That's actually my next slide. So you see that there are several layers of the rods. So those are the lines here are rods, and there are some super long rods. So those are rod cells, and there is another layer. So there are like 3-5 layers of the rod cells. So this is a specialty of deep sea fish, not only of this one, the multiple layers, but he has this like super long ones, and underneath there are the short rods. So it is really morphologically adapted as well. Okay. And yeah, as you said, it's super relevant for the vision. So if you want to understand it, you need to look at it, all of it. So also this.
And they have cones that are all grouped. So more like the macula of primates.
So no, so they don't. Yeah. So it's somewhere now. I don't know where exactly was. I don't think it's on this particular slide. Yeah. Here there is a little zone of cones. Yes. So it's the lower. I go back to the orientation. So here this little zone also has cones on top of it. So, but yes, they are grouped in terms of being together. They are not grouped as in some animals that you have grouped cells, but yeah, they are here. But they're minor.
And the second question is, I remember that I read a few years ago something related to the origin of rods from cones. And I would like to know if you think that the situation in which you saw this high number of rods could be like a time frame during evolution, a sort of picture of this transformation. I don't know if...
Yes, I know perhaps what you mean. So it's actually a great question. Another great question you ask. So the distinction of the cone and rod cells, like as it's been for, I don't know, over a century, like being 2 distinct types, might not be 100% distinct. So there are animals where you have something intermediate. There are animals where you get like the rod cells in the shape of cones or cone cells in the shape of rods. So this is known. And yeah, so we would actually like to get with the single cell also to the point, like, might there be either some intermediate type or is it a continuum? And at least for some things, it looks like it may even be a continuum. But I don't think that these 38 rhodopsins would be some kind of cone to rod thing. It's rather within the rods.
Okay.
But yeah, but mostly when you look from the larva to the adult, there you see the cone to rod, and that's where the questions are. So great question.
Okay, thank you very much.
Hi. So I have another question. Did you also see analyzed lanternfish in your study? And did you see a sort of multiplication of green cones or green opsins in this species?
Yes,
it-
they do have that. We so far didn't go further with the lanternfish too much, but I think in the lanternfish, the story is that they are the highly migratory species, so they are going to the shallow water every night. And the green cone opsins might be used for this kind of crepuscular light, so something intermediate. So even in a bit higher intensity of light, the green cones come in the usage. That's one of the options, even though the adult expression is mostly rods. So I think the green opsins will be rather relevant for the larval phase. So
Okay.
when they are larvae, but we don't, yeah, for that we don't have data.
Okay, later I will show some data related to that. That's
Good,
why I
good,
asked.
yeah.
Hello, great talk. I have a couple of questions by curiosity. First one, you said that unfortunately you didn't manage to observe this fish any fish alive. Regarding the other deep fish that you described, I don't remember their name, but you described that they have mostly rods. In their case, do you know any behavioral activity that they have in common where you can depict the reason of this mutation? And second question is more regarding this spiny fin. Is that possible that is an atypical evolution in order to develop a three-dimensionality visualization, like for every specific wavelength? And thank you.
Yeah. Okay. So I'll answer the first one, which will be easier. So we have some, like we as humans, not we, our group, have some videos or observation of some deep sea species. Some of them we have sequenced in our group and worked with, but it's always just a snapshot. You know, they see it swimming and they film it doing something, but in most of the cases also they use some illumination to see it. So the fish is probably under some stress as doing something unnatural. So I think to be really able to say something on the behavior of these truly deep mesopelagic fish, we need to wait still some time until we really get some observatories or until we get them in the aquaria and we will be able to observe them. Unfortunately, I hope it will happen in my life still, and I believe that it will, because it's now really developing super quickly, but still super quickly means that 10 years ago it was not that many videos as there is now. What they have I'm not sure if there is anything we could draw from the videos, what they have in common to have the same retina composition, but I guess it would just be the shared thing is that in most of the species you get mostly rods or mostly rhodopsin in the retina. That might be simply coming from the fact that they live in the low light conditions, and there will be some kind of default which has evolved in every group because that's easy. So you just get rid of the expression of the other cone cell types and you just keep only the rods, and you have only the rods, and then you do something with the rods because that at least allows you to see something. Yeah, to go into more detail, I think we need to wait, even though there are other groups, or maybe we could talk 5 hours about it, which have some red bioluminescence, red vision, and so on. But that's exception. So the default might be rod-only retina with only rhodopsin and one type of cells. The second question, you ask if this could somehow serve the 3D vision in all color channels. Yeah, I didn't think about that, so maybe it could. I'm just... yeah, I'm just not sure because whatever type of the photoreceptor cells you would have in your eyes, the 3D vision would depend rather on the shape of your eye. So even if you have only one type of rhodopsin, like what the barrel eye has, they have 3D vision because that's binocular, so they're both eyes covering it, and that makes it 3D. So in this guy, it will be also same because the eyes are kind of coming out of the head, so there will be certainly a 3D vision at some field. And then the advantage of seeing everything, like all the light of the spectrum, yes, that would also include the 3-dimensionality just as a default. But I cannot now imagine that this would be the trigger for it to evolve. If I got it right, what do you mean? Is that a bit? Yeah. Okay. Yeah.
More questions? Or there are questions from the audience online?
Okay.
Yes,
Please.
may I?